differences between these major life forms. Right? So eukaryotes, that's the one in the center. It's a diploid genome. We know it's 80S ribosome, 60 plus 40, whereas bacteria would be 70S ribosome, right? 50 plus 30. We know it's a haploid genome. Is that the only genetic material? No, there are extra chromosomal material, and that's typically a plasmid, right? So no nuclear membrane, basically it's called a nucleoid, right? Whereas with eukaryotes, and you all know what viruses are, so I'm going to talk about that. But so these are some of the major differences. We've talked about the kinds of bacteria, we've talked about cocci, we've talked about which one is a curved rod? A curved rod. Is there another one? Vibrio is one of them, one that causes ulcers. Helicobacter pylori. So sometimes these forms are important in giving you a clue as to what the causative organism might be, right? We've talked about cocci, we've talked about gram-positive cocci. We have streptococci in pairs and chains, we have staphylococci in clusters. So of the two, which one is in chains? Is it Strep pyogenes or is it Strep pneumoniae? Pyogenes, very good. And Strep pneumoniae, we usually find it in twos, right? Strep pyogenes beta hemolytic? Yes. Oh, very good. So with some bacteria, we absolutely must have an antibiogram, Staph aureus for instance. And basically what that is, it's a profile of the antibiotics that the bacterium is susceptible to or resistant to. And you all remember that we have charts, right? That tells us whether it is sensitive, intermediate, or resistant. So that is a must for us when we do the antibiograms. Okay? We've talked about peptidoglycan. We've talked about how gram-positives have more thicker layer of peptidoglycan than gram-negatives. What are the differences are there between gram-positives and gram-negatives? Right. So in peptidoglycan, we have the cell wall, and so that's peptidoglycan and just the cell membrane. Whereas with the gram-negatives, there are two membranes, right? There's the cell membrane, there's the cell wall, there's the outer membrane. What kinds of proteins are found in the outer membrane? Lipopolysaccharide, foreign proteins, foreign proteins, lipopolysaccharides. These are all unique things. What else is LPS called? Endotoxin, right? It causes septic shock, and it can cause death. So that's the endotoxin. With gram-positives, we have thyroic acids, lipotyroic acids, etc. So there are differences between the two that allow these bacteria to do things inside the human body. So regardless, both of them have peptidoglycan, and this is composed of linear chains of N-acetylglucosamine and N-acetyluramic acid. You all know these names, these terms now, right? And they're linked by peptides. So they act as bridges between the polymers, and it just adds to the heavy cross-linking that you see in peptidoglycan. The lipid moiety, that's the one responsible for endotoxic shock. Basically, it potently stimulates the immune system, and it results in these other immune players rushing to the scene. Okay? So that's your LPS. We've talked about all of these endospores. Spores are metabolically inert, so they're inactive, and once you give them food, their nutrition, then they germinate and start growing and become vegetative cells, right? So once they start growing, there are ways by which we can measure growth, right? So we've gone through this. How do we measure bacterial growth? Basically, ultimately to look at live cells, we will be doing what's called colony forming units. So that's the CFU per milliliter of growth. So even if you're doing, so basically, you have, what you're going to factor in is the number of discrete colonies that you see times your dilution factor, okay? And you all know this, right? How this is done, we've gone over this before.
Do we have to calculate it?
Pardon me?
Do we have to calculate it?
In this case, it's easy, right? It's everyone knows what 10 times 20 is, right? So that would really help. We've talked about, so basically I'm hitting concepts, okay? When we do measurement of growth, we are measuring the logarithmic phase of growth. That's where active replication is taking place, binary addition is taking place, right? So that's where you see the exponential growth. We've talked about horizontal gene transfer, that's conjugation, transduction, and transformation. Transformation is just free DNA, conjugation is with the use of sex pills, and then transduction is with the use of phage. And we have specialized and generalized. Don't be rude. Okay, but you're not going to prompt me, you're not going to be rude. Okay. Controlling microbial growth, how are we going to do that? What are the different ways in which we can do that? Right? Do you all understand the terms between antisepsis, disinfection, sterilization, etc.? What is sterilization? How are we defining sterilization? Killing all microorganisms. What is the most hardy microorganism? Endospore. So that's our benchmark for finding out if something can sterilize or not, right? Can all things sterilize? No. We've talked about antibiotics, what are the different targets, and I have brought it to your attention when we talked about different specific bacteria and what kinds of disease they cause, whether obviously all of the bacteria will have antibiotics. If I've not included them, it's because there were too many, so then it would mean appropriate antibiotic based on what else is going on. But in cases where I've given you a list of antibiotics, I do want you to know them. And we have gone over the mode of action for them, in which case, please know what they are, okay? So we've talked about cell wall synthesis, whether it punches holes in the plasma membrane, whether it interferes with nucleic acid synthesis, ribosomal, whether it's 30S or 50S, ultimately it will block protein synthesis, and that, of course, is going to prevent the bacteria from growing. Or we talked about folic acid for the bacteria to prevent folic acid synthesis, right? We have drugs that can do that also. Can anyone tell me what the two drugs are that we've talked about that does that? Trimethoprim, sulfamethoxazole, right? They both will target folic acid synthesis. Now, when you put a person on antibiotics, it's also going to wipe out the whole flora, right? So we talked about pseudomembranous colitis. Which organism causes that? C. diff, Clostridium difficile. Is that anaerobic? Yes. Is that part of the normal flora? Yes. In everyone? No, just in some individuals, right? So when that person is on what kind of an extent, what kind of a drug do you see the Clostridium broad spectrum variant? Okay? So normal flora has a function, right? We've talked about this. We've talked about the significance of normal flora. We'll come to that in just a second. But these are the sites, so basically everything that's open to the exterior, right? Those are colonized by bacteria, viruses, fungi, protozoa, whatever it may be. But it's a mixed community, right? But not all sites will support normal flora. There are some that are sterile, right? Blood is considered to be sterile. Your bones, sinuses, etc. These are considered to be sterile. That means no bacteria or viruses. So if we find organisms in these sites, and that's indicative of an infection, and that has to be treated, you have to medically intervene and flush out those organisms. So these are the benefits of normal flora in your body. Okay, so they're going to basically compete for nutrients, compete for binding sites. So the pathogen cannot establish a total. They can produce bacteriocytes, which are going to kill other bacteria. So because it's a competitive environment, they're going to try and establish themselves. Right? So they can be very helpful to us. Some of them secrete vitamins that help us. It can also cause disease, as you know. Some of the bacteria we've talked about, like Haemophilus, Strep, Staph, etc. These are what we call opportunistic pathogens. That means they hang out in the normal flora, but then when conditions are primed for it, or if they're introduced into a location that is normally sterile, now they have the opportunity to cause disease, and that is because they have virulence factors. And what are virulence factors? It's something that will help the organism to cause disease, right, in that new environment. Okay. So types of virulence factors, we've talked about them. They promote colonization, they can help survive, they can help avoid the immune system, they can actually help spread, go beyond the initial site and go deeper. They can hyaluronidase, we've talked about, coagulase, coagulinase. All of these are spreading factors. They help the organism go beyond the site of entry, right? And when we talked about the individual bacteria, we did talk about those also. There are some that will help inhibit phagocytosis, for instance, capsules. They're slippery and they won't even allow the phagocyte to engulf the bacteria that have capsules. There are others that will kill the phagocytes with toxins. There are some that degrade the complement protein. By doing that, it will prevent this particular protein will signal neutrophils and make them come to the site of injury. But if you cleave it, then it's not going to do that. So several different ways bacteria can do this, right? Some will escape the phagosome before it even fuses with the lysosome, right? This is Listeria. When we talked about monocytogenes, we talked about this. Some prevent the phagosome-lysosome fusion. So there are different ways that the bacteria can help can survive, okay? And try to avoid what you call it. We talked about M protein, which is a virulence factor because it prevents binding of complement proteins. So do all bacteria have the M protein? No. So Strep pyogenes was one of the ones we talked about. Now, what is one of the common infections you see with Strep pyogenes? Strep throat, very good. Right. So what are the sequelae with strep throat? Rheumatic fever, nephritis. So you see that because, and basically that's an autoimmune reaction, and you see that because of cross-reactivity. The antibodies that are made to M protein will also cross-react with some of the cell protein. LPS, your endotoxin, will bind sialic acid to prevent the C3 convertase, which is important in the complement cascade. So you learned about complement from Dr. Demery Butler. Have you started complement already? Okay, so you'll see it's a cascade, the ultimate goal being destruction of whatever it's focused on, right, through that membrane complex. So these are some key enzymes. So by inhibiting that, you prevent that cascade and you prevent the ultimate destruction. Bacteria can also lengthen the O antigen, which is the one that's outermost on the LPS, right? It starts with lipid A. So O antigen, you can lengthen that to prevent that membrane attack complex, which is basically initiated by the complement. Which bacterium did we talk about that has urease? That I'll give you a hint. Yes, Helicobacter pylori. What other virulence factors does Helicobacter have? Glycinase, flagella, dactylating cytotoxin. Very good. So you're on top of things. Very good. Siderophores, basically these are chemicals that are going to bind iron, and bacteria need iron. Bacteria will need iron to grow, and they capture it from their environment. So siderophores are just these chemicals that are made to bind iron and bring it in. Capsules, we already talked about. Protein A, Staph aureus, right? So what does protein A do? It's going to bind to the Fc portion of the antibody, and it prevents oxidation. So we've talked about this several times. One last time, so this is your protein A, and it's going to bind to the Fc portion of the antibody. By doing that, it prevents it from binding to these Fc receptors on the backside. That means there is no oxidation. Otherwise, what happens? The antibody binds to this antigen, the Fc portion of the top, it binds to the Fc receptor, and then the entire thing is taken into the phagocyte. That is basically, it's eating, right? It's eating that germ, it's taking it inside. So that doesn't happen, the bacteria continues to grow. Okay? So that is proteinase. So proteinase, Staph aureus, M protein, strep pyogenes, right? Mucinase and urease, flagellum. We talked about Bacillus you need to be Helicobacter, but these are virulence factors that we're talking about for different bacteria that cause human disease. Okay? So bacteria can also bind to host proteins or polysaccharides to make themselves appear as self-antigen. Antigenic variation, this is not new just to bacteria, right? We've seen that also with viruses. Okay. There is the secretory IgA protease, the S-IgA protease, which will basically cleave it at the hinge region, and it will, the end result being the same as protein A, there is no oxidation. Okay. Some will reduce MHC class II molecules on the surface of the host cells, which that is an important part of the immune system to do antigen presentation, because when these antigens are chopped up, they are presented in context of MHC class II. So if you reduce that, you're going to see less of that antigen presentation. We've talked about spreading factors, we've talked about toxins, we have exotoxins as well as endotoxins. Your endotoxin is your LPS. And exotoxins are toxins that are secreted, right? So when we've talked about exotoxins, we've talked about the tetanus toxin, right? Do we have a vaccine against tetanus? Yes. Is that important to know? Yes. When can it be? We've talked about the earthquake, right? So if you have a patient that you have no records on, right, and has exposure to soil, he's buried under all that rubble, etc., then that is something to keep in mind. It's an easy thing to do to give a tetanus booster and potentially prevent tetanus, right? So we have several vaccines against tetanus. We have botulinum toxin, right, which is also secreted. They both show paralysis. Do they show the same kinds of paralysis? No. What is the difference? Flaccid and spastic. Which one is flaccid? Botulinum. I'm so proud. Exotoxins, there are several different modes of action, right? And depending on what the end result is, we can call them out, right? We have membrane disrupting exotoxins that will punch holes in the membrane. We have the AB, where A is the activity domain and B is the binding domain, so they act together. So B will bring it into the cell and then A does the activity. We've talked about superantigens. How do superantigens work? They indiscriminately activate large numbers of T cells. And how do they do that? So this is what your normal activation is, right? So this happens in about 1 in 10,000 T cells. A superantigen will not wait for this specific reaction to take place. A superantigen will bring the two together by binding to the antigen-presenting cell and then also by binding to the T cell receptor. By doing that, the number of cells that are activated goes up considerably, from 1 in 10,000 to 1 in 5 T cells. Imagine the huge flare-up of the immune response, right? So what happens? Up to 1 in 5 T cells are activated, and it results in excessive production of immune mediators, right? So these cytokines, what they are going to do is they're going to attract more cells to the scene of injury, right? So this also activates your self-reactive T cells, and that is not something we want. So when this happens, when you see this over-exaggerated immune response, ultimately what happens is you see an immune suppression, right? Because these activated T cells are ultimately undergo apoptosis. That means programmed cell death. That means the body will get rid of them. They're going to die, right? So that creates a big deficit then. That creates an immune suppression, generalized immune suppression, which is not a good thing, right? So this could then create septic shock. There could be multiple organs in the body will start failing and ultimately result in death. Okay, so this is how superantigens will help the bacteria overwhelm the body's immune system and ultimately take down the system. So when you have an organism that has a superantigen as your etiologic agent, you have to provide that medical help. You have to provide the drugs, and you have to provide it in a timely fashion. So we talked about getting antivirograms. We know we can do MIC. You all know what MIC is, right? It is the minimum inhibitory concentration of the drug, right? So we can do it in two ways. We can also look for sensitivity that we can do in two ways. The Kirby-Bauer method or the disc diffusion method. So the disc has a known concentration, one concentration of the drug, and you put it on an outer plate with the bacterium, and then basically you measure the zone of inhibition, right? This diameter, that's your zone of inhibition. And then you compare that to a chart, and then you have your E-test, right? So your E-test is a strip of paper that has a gradient, that has a varying concentration of the same drug. And then your MIC is where the two meet, right? This clearance meets the paper, okay? So that becomes your MIC. So we'll talk about a few of the infections today. What do you know about impetigo? Which organism is it? Very good. What else do you know? It's the most common bacterial infection in children, right? And it spreads easily from one part of the body to another, so it is contagious. What is treatment? Mupirocin, 2%. It's a topical treatment, right? Avoid close contact with the patient, changing clothing, bedding, washing it separately from other items, etc. Typically, the children are kept home from school because it is a highly contagious infection, right? Another thing that Staph aureus can cause is scarlet skin syndrome. Can this be potentially lethal? Yes, so you have to intervene. This is a potentially fatal toxin-mediated disease. So the demographic, look at the demographic. They have such an underdeveloped immune system as well, right? So when you provide help, it has to be immediately. Treatment should begin immediately. You suspect it, you should treat. So typically you give them beta-lactamase resistant penicillin, so methicillin, ampicillin, etc. These are your beta-lactamase penicillins. You can also give them first-generation cephalosporins. So that's your scarlet skin syndrome, gas gangrene. Which organism causes it? Clostridium perfringens. So two conditions must exist. Now this is an anaerobic bacterium, right? So there has to be an anaerobic condition for the spore to germinate, to start growing, and then create the problem, right? So it causes a very strong foul odor. It creates a lot of gas. That's why it's called gas gangrene. There may be gas bubbles that would actually, it would distort the tissue, contributes to necrosis. And the gas bubbles you may actually be able to cure it. Those snap, crackle, pop as the patient is repositioned. Okay? How do we treat gas gangrene? That becomes important. If you don't do this, if you don't do all of this, you may end up having to amputate a limb so that you can save the rest of the patient, right? So wound debridement becomes a critical component of how you're going to care for the patient. That you're getting rid of the necrotic tissue, because necrotic tissue is going to facilitate more growth of this bacterium. So you're getting rid of all the dead tissue and cleaning out the wound, etc. You can do hyperbaric oxygen therapy. So basically you're infusing oxygen into the tissue, thereby trying to kill an obligate anaerobe, such as Clostridium perfringens, right? And when you give them the drugs, it is penicillin and clindamycin together. Okay. If you don't do that in a timely fashion, then you end up having to amputate. We talked about necrotizing fasciitis, Strep pyogenes and Staph aureus that can both cause this flesh-eating disease, right? So basically the subcutaneous fascia and the fatty tissues are destroyed by the growth of these bacteria. Okay. So Strep pyogenes, we've talked about this already, gram-positive cocci chains. Is there a vaccine for Strep pyogenes? No, unfortunately we don't, but we do have vaccines for Strep pneumoniae, right? So So what are the virulence factors for strep pyogenes? It is capsulated. We've talked about how it has M protein. It is a virulence factor. How does it function? It destabilizes the complement component, which is called C3b. Okay, so if you get rid of this, then it's not going to allow the cascade to take place. The ultimate end result of the cascade is that membrane attack complex formation. So it won't allow that to take place. Okay? It also has a C5a peptidase, which will degrade the C5a complement component, so neutrophils cannot migrate to the site of infection. And it has your spreading factor, right? We have hyaluronidase. What is hyaluronidase? What does it do? It gets rid of that glue between the cells, so it can go deeper past the initial site. Okay? It's got streptolysins, it's got streptokinase. We've talked about how these function. It also has superantigens, and we know how superantigens function, right? So these are the streptococcal pyrogenic exotoxins, right? That result in that overexaggerated inflammatory response followed then by that generalized immunosuppression, which will then allow the bacterium to keep on growing, which could ultimately result in death. Okay? So strep pyogenes, we've talked about catalase positive or catalase negative for just a few of the bacteria. Can you give me an example of a bacterium that is catalase positive? Staph aureus, very good. So they're both cocci, so catalase negative would be streptococci, and catalase positive would be your Staph aureus. Okay. Trachoma is an eye infection. Which one causes it? Chlamydia trachomatis. Now, it's a serotype that causes trachoma, but that serotype will not cause sexually transmitted infections. Okay? So, trachoma can also be transmitted by mechanical vectors. So flies would be an example of that, or contaminated fingers. So don't have good hand hygiene, etc. Children can transfer it to each other, so that can also happen. Okay? Chlamydia is neither gram-positive nor gram-negative. They have an atypical peptidoglycan that lacks N-acetyluramic acid. Okay, so it has N-acetylglucosamine, but not N-acetyluramic acid. Unfortunately, I can't go over everything, but I'm going over what I think are some key aspects. We talked about bacterial meningitis. Not all of these we have vaccines for, but we do have vaccines for some of them. And it may be the demographic that may be more at risk for one versus the other. So in adults, it's strep pneumo and Neisseria meningitidis, whereas in children, it's Haemophilus influenzae type B and Neisseria meningitidis. And newborns, you have strep A-lactate and E. coli. You also see Listeria monocytogenes, but this is less common, and if found, it's usually found in adults or newborns. Okay? So we know there are vaccines. The vaccines are your Haemophilus influenzae type B vaccine. So what age group do we give it to is 2, 4, and 12 months of age. Okay? So that's your HIB vaccine. With Streptococcus pneumoniae, we have a 23-valent and a 13-valent vaccine, right? So know the vaccine, know the age group that we give it to, and there are so few of them that we talk about that has a vaccine. So if we do talk about it, please know that it is available and who we can give it to. So in this case, we talked about Haemophilus influenzae. Does it typically, it affects children, right? Under what age group, do you remember? 5. Very good. Look at you, how proud I am. Very good. Okay. We have Neisseria meningitidis vaccine also. So these are your gram-negative, they're diplococci, they're kidney bean shaped, right? So this is your Neisseria meningitidis. It causes the most serious form of not just acute meningitis, but also your epidemic meningitis that leads outbreaks. Okay? So in schools, etc., that could be an issue. So what are some of the virulence factors? It has a capsule, so it allows the the bacterium to escape phagocytosis, but it also has IgA protease. And what is the ultimate end result for IgA protease? No opsonization. It's going to cleave the IgA, right? So there is no opsonization. Haemophilus influenzae type B rarely infects over the age of 5, so it's mostly an issue in children and infants. Not newborns. In newborns, it's E. coli and strep Agalactiae. Okay? So the vaccine has been immensely helpful. In this case, the virulence factors, just like Neisseria meningitidis, the capsule and IgA protease, but in addition, some of them also produce a beta macromoles. Tetanus, the spores are found in the soil. The best example to help you remember is that earthquake. So anything that potentially gives you trauma and exposes you to soil could introduce this into the person, right? So the spores enter deeper tissues where the conditions are anaerobic. This is an obligated anaerobe. Once the bacterium starts germinating, then it can produce its toxin, the tetanus toxin, which will then create tetanus. Okay? Causes spastic paralysis as opposed to classic paralysis, which the botulinum toxin. The good news is we do have post-exposure prophylaxis, right? So we can do prevention, that is the booster dose every 5 to 10 years, and then we also have anti-toxin. Botulinum, what is a demographic that this could be a problem for? Infants, right? Infant botulism. How are you going to prevent that? Are there certain foods that you should not be eating? Honey and corn syrup. So why these? Because they may have spores. If we ingest them, our gut flora is well established that it will outcompete. It won't allow these bacteria to grow. But in an infant, a newborn, or a young infant by the age of 1, the gut flora is still trying to develop. So it's not fully established yet. So then can potentially cause an issue. So it causes spastic paralysis, and treatment would be your trivalent antitoxin. We've already talked about this. This is the most potent bacterial toxin known, and it helps us because then when we think of therapeutic approaches using this toxin, we can figure out how little to use it and what we want it to do. So we've talked about this.
Professor, I have a question. Is that treatment for infants or for adults?
Infants.
Oh,
Right? Yeah. In adults, why doesn't it cause an issue in adults? Because our normal flora is dominant. Okay, so endocarditis, we talked about acute endocarditis and subacute endocarditis. Basically, it's the length of time that it takes for the symptoms to be obvious to that person. Staph aureus is an issue, especially in IV drug users. You can also see strep pneumo or strep pyogenes, and also Neisseria gonorrhea that can be etiologic agents. What is bacteremia? It's presence of bacteria in the blood, right? So some of the most common agents are Haemophilus influenzae type B. So where else did this come up for us? Meningitis. Meningitis in children under the age of 5. How are we going to prevent that? Vaccine. The same with Neisseria meningitidis, we have a vaccine. The same with strep pneumoniae, we have a vaccine against 23, depending on the age group, etc. We have a 13-valent and a 23-valent vaccine. Salmonella enterica or Listeria monocytogenes. So all of these can cause bacteremia. That means they slip into the bloodstream and then potentially cause a more serious infection. Okay? With Listeria monocytogenes, some of the virulence factors that we talked about that help to cause disease, one is internally. It forces the cell to phagocyte the bacterium, right? And then it spreads. So once it does that, right, into a non-phagocytic cell, then it uses this actin polymerization to spread from cell to cell. What is the advantage to the bacterium in not going back outside? It's not avoid the immune system. Very good. Listeria lichen O will allow it to escape from the phagosome, before it fuses with the lysosome. Okay? Lysosome is what's inside the phagocytic cell, and it is filled with toxins. It's filled with poisons that are going to chop up the bacteria. So by doing this, it avoids being chopped up into pieces. Okay? Okay. Do you have any questions for me? So it's 2, 4, 6, and 12, 15 months of age. You're welcome. The Haemophilus influenzae type B, the vaccine, 2, 4, 6, and 12 to 15 months of age. So 6 months of age is if needed, depending on the brand of the vaccine, and then the final booster dose. Okay, so there are several different formulations. So for all of them, 2, 4, and 12, 15 months of age. But in some of them, depending on the brand, it's also 6 months of age. Okay, this is your Haemophilus influenzae type B. And children are the demographic that we worry about because that's the demographic that mostly causes the problem. Which organisms cause issues with otitis media? Haemophilus and strep pneumoniae. What kind of Haemophilus? Type B, non-typable? Non-typable. What's that mean, non-typable? What is it? No capsule, right? So how do you type them into different serotypes based on capsules? So that would be capsule or polysaccharides, right? That's how we do the serotype.
Professor, I have a question. Why is HIV part of a normal flow?
Why is
HIV part of a normal flow?
HIV?
I mean HIV.
Oh, why is it?
Yeah.
Good. Okay. So how is normal flora established, right? So let's talk about a healthy person. In a healthy person, a healthy individual may even have MRSA, right? In their nostril, perhaps in their nose, right? But it's an opportunistic pathogen. It is there because it can be there. It does not get out-competed. With the help of its Vibrio and with its ability to use certain nutrients, it is able to grow in that environment. But it doesn't cause disease because it doesn't go past that initial site of entry because everything else is functioning. the person's immune system is functioning, there are no other diseases, there isn't any trauma, etc. So any of these, including Haemophilus influenzae, any of these opportunistic pathogens can cause disease when that opportunity presents itself. And that opportunity may be an accident, so trauma, considerable trauma. So all of a sudden, these organisms that are parts of the body that it cannot go beyond, all of a sudden now find themselves in the lung, deeper in the lung, or in the bloodstream, able to cause the blood-brain barrier, set up shop in the brain. The brain is sterile, right? So that it causes disease. So all it needs is an opportunity. So the reason it is part of normal flora is because it can be there without causing disease in an otherwise intact and healthy host. So it's basically evolution. So it's basically evolution, right? So we evolve over time. You have this happy good. Caleb.
You mentioned the lung being having normal flora, but then I think on another slide you said it was sterile. I wasn't sure if the lungs were sterile or they're.
Right. So the lungs are you have your upper respiratory tract and your lower respiratory tract. So part of the lungs that are considered to be sterile are your bronchioles, which are at the very end where the gas exchange will take place. Lydia?
I know that Haemophilus influenza B has beta-lactamase. Is it safe to assume that the Lomidalva capsule is also has beta-lactamase?
Yes. So how do we treat something that has beta-lactamases? Beta-lactamase resistant penicillins. Do we have beta-lactamase inhibitors?
Yes.
What are
And something with back, more the back.
Kendall, did you have a question?
Yes, Dr. Martin. Regarding the antibiotics, when you are going to ask about it, are you going to ask us based on the disease or based, for example, erythromycin? When you look there, when you went over it, it tells you all the diseases that it can cure. Do you want us to know all of them, or do you want us to know like specifically for which disease was it when you went over the disease?
So, yes. So with the antibiotics, the kinds of questions I have asked are which antibiotics for a certain disease, for instance. Okay? So that is one way to use the knowledge. The other one is the mode of action. So we talk about some antibiotics, not all of them in class, so then know what the mode of action is for that particular antibiotic. How do the macrolide antibiotics work? How do the tetracyclines work? How does concentration work? Things like that. How does ciprofloxacin, how do quinolones work? Right? So know the mode of action for each drug. I've tried my best to keep it fair. In fact, I have kept it fair, and I've tried my best to keep it simple, but I don't have the best perspective for that.
We have a question in Palm Beach.
Yes.
What should we focus on for the last two lectures? I realize we didn't have time to go over it, but there's a lot of material on there. Is there anything specifically we should be looking at?
All of
The
it.
sexually transmitted bacteria.
All of it, because what I've done is really narrowed down my focus, and I've only focused on what I thought were very key factors for each of those bacteria that we covered towards the end.
Thank you.
So do well. You look ready, and I'm so happy to see that. So just keep doing what you've been doing. Give it one last push.
Is it just the gastroenteritis?
Okay. So the question is, we all know ones that aren't part of the normal flora are the enteropathogenic E. coli, the enterouropathogenic or the aggregated, all of those ones that have that extra designation. Those are the ones that are not part of the normal flora. So where do they come from? They come from, they are the guts of other animals. We talked about some salmonella that can be found on reptiles, so turtles, etc. Which one was that?
Salmonella.
Which one?
Salmonella.
Salmonella, which one?
Oh.
Enterica. What about the other salmonella? Typhi? Do we find that in other animals? No, just humans, right? What about Shigella? Also humans. What about, we talked about Helicobacter.
Dr. Miley?
Yes. Yeah.
Yes, I have a question on this. I just want to be clarified on the cephalosporin medications. Do we need to know all the drugs for the generations, or do we know only
If
the
I have
specific generation what it are?
Right, if I have mentioned it, please know it.
Okay, so just the one on the PowerPoints.
What is the other question? Sorry, should I?
Professor,
Yeah?
I have a question here. Maybe here.
Yeah? Who is it? Okay.
No, it's just looking. Okay. The only bacteria that causes the scaly phylosome lysosome is the Listeria monocyte, right?
Listeria monocytogenes.
It's the only one, right? I cannot remember.
Which one? So they both ultimately escape phagocytes. One does it by escaping the phagosome and one preventing the fusion. What are they? Listeria and Legionella. What does Legionella cause? Pneumonia, legionellosis. Where is it found? Water, warm water. So that would be found. So typically not all ACs or typically not all ACs. It's not an issue. Which is the most fatal tick-borne? Very good. Which organism is it? Rickettsia. Is there something special about its cell wall? Yes. Does it have? What does it not have? No. It has energy? It does not have it. Is there a tick-borne disease where you have to give treatment immediately?
Oh, yes. The Rocky Mountain.
Rocky Mountain? What about Lyme disease?
Lyme.
And Rocky Mountain is the most fatal, so what is that cutoff point? 5 days. If you want to do it before the petechiae, the spots, look here. Do you know which antibiotic? Yes, we should love to know. So, what I have done is, the kinds of questions will be connecting pieces of information. If it's too simple, it is too simple. You all have the information, and we have more time. It's 2 points per question. There are 50 questions. So just be calm, be cool, and reason it out, right? Reason it out in your head. By process of elimination, some will be obviously wrong, right? We talked about certain virulence factors. Obviously, not all bacteria have all the virulence factors we talked about. Some of the big ones are M protein, protein A, the superantigens, the SPE superantigens, which bacteria have those. So pay attention to those kinds of things. Those are your hints, right, in the question. We've talked about so few of them, we talk about actual antibiotics that we are using. So when I mentioned that, and there is a potential urgency to giving it to the patient, please know that, okay? If you don't have any questions, I wish you good luck. I want you all to do well.
One last question, we were talking about toxic charges, antigens, we were talking about careers and so we were talking about the TSST1, the antigen for staphylococcus and the SPE for Pyogenes. They also cause toxic shock, right? Okay.
So super antigen